Okay, we're back with David Sanders, a professor of virology at Purdue University. And David, we're on to part 4 of our mini-series of taxonomy of viruses. And now, this time, you're going to talk about retroviruses.
Yes. So retroviruses are not called retroviruses because they dress like they're in the 70s. They're called retroviruses because they were perceived as doing something backwards. And I'll tell you about that in a moment. I will just, at this time, assert to you that, in fact, they aren't doing it backwards. It was our concept of the nature of the flow of genetic material that was backwards. It's not the viruses that were backwards. But in any case, they originally called RNA tumor viruses because they were associated with cancers in animals. The best known retrovirus, which is in fact belongs to a class of retroviruses called lentiviruses, is the human immunodeficiency virus, HIV. But let's talk about a little bit about retroviruses in general. So retroviruses, they're the genetic material which is present in a retrovirus particle is RNA. So there's RNA that is present in the particle. But there's another RNA which is also present, and that is called a tRNA or a transfer RNA. And that is incorporated into the virus particle. So there's an RNA that actually encodes the viral proteins, and then there's this tRNA. As it happens in retroviruses, there's an additional feature. There is, in fact, a second copy of the RNA. So it is what's called diploid. What is that, diploid? Well, you and I, Ellie, are diploid. That is, for each one of the chromosomes that we have 2 copies of it. A bacterium, when it's not replicating, is monoploid. It just has a single chromosome. But animals, again, generalizing here, certainly mammals, certainly you and I, are diploid. We have 2 copies of each of the chromosomes. Of course, the X and the Y chromosome, for men, they aren't exactly the same chromosome, but they form a pair. In any case, this virus is also diploid. Now, that's very important for it and how it replicates. But for today, just to eliminate the confusion, I'm just going to get rid of it because we're not going to focus on what happens with it. So this is what comes in. And what's interesting about retroviruses is that even though the genetic material in the virus is RNA, and I'll just as an aside indicate that it could be that there's a little bit of DNA present as well. But what's important is that the retrovirus in its life cycle will have RNA when it's outside the cell in a virus particle, but DNA when it is inside a cell. So it has both of those forms. So what happens is the activity, the protein, the enzyme that's necessary to do those processes doesn't really exist per se in host cells. I'm cheating a little bit there because it sort of exists, but in any case, the virus won't be able to readily access it. And you may recall in our discussion before about RNA viruses, where we talked about the positive sense RNA viruses, the ones that have a positive stranded RNA viruses, that their viral message, when it enters the cell, is translated into the viral proteins, including the protein that's necessary to replicate the RNA. So that's for the positive strand, positive sense RNA viruses. For the negative sense ones, that RNA can't be translated into proteins, viral proteins. So in order to generate the positive strand, they have to bring in with them the replicase protein, the RNA-dependent RNA polymerase. And that's necessary to make the positive strand, which can then be translated into the viral proteins. It turns out that for the retroviruses, the same thing is true. They have to bring in the protein, which is the what's called the reverse transcriptase. So they have to bring that in with them into the cell. So what happens is that it turns out that we have this little bit of double-stranded RNA because the tRNA is bound to a complementary sequence in the mRNA. And what happens from this is that we generate a DNA molecule. And again, I'm simplifying the process greatly, but this enzyme copies this and makes DNA. So it started off with an RNA template, but it makes DNA. And then we wind up, again, I'm simplifying this, we wind up getting rid of this, we wind up with a full strand, and then this same enzyme makes a second strand of DNA, and this RNA goes away. It's degraded. Turns out that this enzyme both makes the new DNA and degrades. There are two components to the enzyme. Also degrades the RNA, so it gets rid of the RNA. So we're left with a double-stranded DNA molecule.
That's two strands of DNA.
Two strands of DNA. So we had a very interesting set of reactions here. We had a DNA being made using RNA as the template, right? So we're making a complementary DNA to the RNA. That's reverse transcription per se. And we were using RNA as the primer. RNA is what got that process started. The tRNA got that started. Then we made a second strand with we made DNA. So it's a DNA polymerase using a DNA template. So
is that an identical copy?
So this sequence would be the same as the original RNA, right? So this is the complement, as we described before, because of the base pairing and so on. So this is a complement, and then this one now becomes the template for this. So this has the same sequence as this, with the one little exception that in RNA we have A, G, C, and U, whereas in DNA we have A, G, C, and T. So the sequence of nucleotides in this would be exactly the same as this, except the U's in this will now become T's in this. Now, once again, I am simplifying because it turns out that the ends of the DNA wind up having additional material. They have a duplication which occurs in this process because of how the process occurs with primers, with these RNA primers, and how the enzyme moves around. It winds up that this, if we put a little box around this and this, we'll put this, maybe make this a little different. It doesn't really matter. It's a bit complicated, but these boxes wind up getting duplicated. So we would have this present at this end and at this end. So what's originally at this end and this end winds up together, and it's duplicated at the two ends.
Okay.
And we understand that process very well, but I don't think it's worth spending a lot of time explaining exactly how that happens. But that's how it happens. All right. So we no longer have the RNA. We have this thing here, and I should point out it's also present here as well. So now we have this DNA. So what? What are we going to do with that DNA? Well, that DNA has to get into the nucleus of a cell. So we're inside it. This all has occurred. All this replication has occurred inside the cell. So it's possible that it just got started a little bit inside the virus particle, but that's possible. People disagree about that, but this has to get into the nucleus, and when it gets into the nucleus, there are chromosomes, right? And what happens is that this same enzyme, there are actually three parts of this enzyme. Again, I'm simplifying here. There's three parts of the enzyme, one of which makes the new DNA, one of which destroys the RNA, and the third part of it actually takes this. Now it's working, again, I'm simplifying here, but it takes this part and it gets incorporated It's not eliminating this part, okay? It's just adding to this part. But if we so instead of showing this as a chromosome, let's show this as two strands. So it winds up that after insertion and replication, it winds up that we have the DNA from the retrovirus inserted into the host cellular nucleic acid, genetic material. So a retrovirus is forever. Once it's inserted into the DNA, it becomes a permanent part of the genetic material, and it is in that cell, in the nucleus of that cell, and its daughter cells, because as this cell divides, that will also be transmitted to daughter cells as long as they exist.
So just so I understand, once the DNA has inserted itself into the host cell DNA, how are new particles of virus created?
So that's a great question. So this DNA is mostly, and I'm trying to be careful here, is mostly not recognized as anything but now cellular DNA. So I should point out that cells do have ways of regulating retroviruses and potentially seeing that this is not quite its DNA and trying to shut it down and so on. But frequently, most of the time, much of the time, retroviruses are successful in just having their DNA look like the rest of the cellular DNA. There's no real way for the host to discriminate it. Again, I'm simplifying here. So normally what happens with the DNA that is active is that RNA molecules are being made from them, and then they leave the nucleus and they get translated into proteins, the ones that encode proteins. They get translated into proteins, and those proteins go off and do whatever they do. So that's the product of this process. The same thing is true for this material. So this material will also make, in this case, it will make the messenger RNA, the mRNA of the virus. And so again, simplifying because it can be quite complicated, but this once again will go outside, interact with the ribosome, and we will make the viral proteins. The viral proteins include this protein, includes the protein that's responsible for making the capsid for the virus. And in the case of retroviruses, it also is responsible for making the protein that goes on the surface and allows it to attach to the next cell and to fuse the viral and cellular membranes. What's neat about the retroviruses is that this same message, which is used to be translated into viral proteins, at least the full-length message, gets incorporated in here. So the exact same message, which is used, again, I'm simplifying here, but that's used to make these proteins and the reverse transcriptase, the protein that's responsible for making the DNA, and the other components. Those other components are referred to as RNase H and integrase. The integrase was the one that got it in here, but they're all part of a larger protein. Those are all made by, again, in general, the same mRNA, the same full-length mRNA that gets into the package. Some of the other proteins, for example, this one, is not made from this mRNA, but a different mRNA. But the point is that the genome is used both for being translated into viral proteins and to being incorporated into the retrovirus. And then this can leave the cell by going, this virus gets made. Different viruses may differ about where exactly it gets into the pathway to leave the cell. Does it happen at the cell surface? Does it happen earlier on? But in any case, this can go on now to the next cell. So there are a few important points here. Number one is, as I said, this DNA is permanently incorporated and looks like the DNA in the cell. Now, it's again, there are things that cells do to potentially limit the amount of virus and so on and so forth. But I think that we need to think about this in a different way from the way people have thought about it in the past. And that is that it's not really true that retroviruses are like RNA viruses. RNA viruses really want to just use the cell and make more of themselves. With the retroviruses, in fact, they are contributing to the genetic material of the cell and actually contributing things that are potentially of value for the evolution of the cell. Remember, I was telling you about these boxes here at the ends? Well, these boxes at the ends have material that can like turn on the DNA or turn off the DNA or have the DNA have, and what do I mean by turn on and turn off? It's how are they producing these mRNAs? And these things can be very valuable for the cell to be able to turn on and turn off its DNA so it's producing mRNAs in a regulated fashion. So that's a valuable thing. It can also, the insertion of this material can also provide the genetic variation that promotes evolution. Just by inserting into a part of the genome, it can cause mutations, can promote mutations, and those mutations can affect other processes that are important. So these viruses, we have to think of them as more, they're not really, they are actually part of metazoan, that is multicellular evolution. I would argue that they are important for the evolution of all DNA-based life. That, again, if we think of RNA viruses as reflections of the RNA world and of RNA-dependent RNA polymerization as being the first sort of nucleic acid polymerization reaction, we might think of retroviruses as being the earliest form of polymerization of DNA. And so, yeah, there's a lot more to be said about that. But I think we need to have a broader idea of viruses and what they're doing. And finally, we have to when we think about why was it so difficult, and why do we still not, for example, have a HIV vaccine? We have HIV antivirals, which are one of the amazing most amazing discoveries humankind has ever made. And it's stunning that people can live a long lifespan with HIV nowadays because of those drugs, but we don't have a vaccine. And the probable reason is that it's a retrovirus. Retroviruses aren't really seen as something totally foreign to us. And this particular retrovirus, HIV, came from a very closely related evolutionary animal, that is, chimpanzees. HIV-1 came from a simian immunodeficiency virus that was present in chimpanzees, transferred to humans probably in the bushmeat trade. So the fact that it came from a very similar organism, that it had at least partially co-evolved with, how long is a question in the scientific community, how long it evolved with the chimpanzees. But nevertheless, these simian immunodeficiency viruses had evolved with primates for a long period of time. And so therefore, it's not that surprising that given that it's a retrovirus and given that it came from a closely related species, that we wouldn't necessarily be able to generate an effective immune response or an effective vaccine.
That's very interesting. Yeah. Thank you, David. That's been great. And we'll catch up next time to talk about other types and aspects of viruses.
Thank you very much.
Thanks.